How do you know if your cells are happy? You look at them. Every expert cell biologist develops an intuition for cellular health, and it all starts at the microscope. No lab? No problem. This episode gives you the complete visual vocabulary you need. Why every cell biologist needs microscopy. Imagine you are managing a team of workers you cannot speak to. The only way you can assess their well-being, productivity, and health is by looking at them through a window. That is exactly the relationship you have with your cells. Cell culture is fundamentally a visual discipline. Before you ever run a Western blot, before you quantify RNA, before you process any downstream assay, you must first know that your cells are healthy, at the right density, and free from contamination. None of that information is available to you without a microscope. Experienced cell biologists can look at a flask for 30 seconds and tell you the confluency, the health status, whether contamination is present, and whether the cells are ready for passaging. That expertise is not magic, it is a trained visual vocabulary. This episode gives you that vocabulary. What you will learn in this episode: the 3 types of microscopy used in cell culture, and when to use each. How to assess confluency, morphology, and cellular stress at a glance. What healthy, stressed, dead, and contaminated cells actually look like. The 4 most common microscopy mistakes, and exactly how to fix them, how AI image analysis is automating this exact process in pharmaceutical labs. If you have ever said, "The cells looked fine," and written nothing else in your lab notebook, this episode is your intervention. The 3 microscopy types in cell culture. Not all microscopes are created equal. In the cell culture world, 3 types dominate, each with a specific purpose, strength, and ideal use case. Understanding when to reach for each one is the difference between a quick, confident assessment and 20 minutes of confused squinting. Brightfield is the simplest form of optical microscopy. White light passes directly through the sample from below, or above, in inverted scopes, and whatever the cells do not absorb appears bright. The cells themselves appear as gray or slightly translucent shapes against a bright background. What you can see: cell shape and approximate size. Gross confluency estimation: whether cells have detached from the surface. Large organelles in highly pigmented cells. What you cannot see: internal structures, nucleus, nucleolus, cytoplasmic organelles, fine details of cell morphology. Early signs of stress before they become extreme. Phase contrast microscopy, the gold standard. Phase contrast is the workhorse of cell culture microscopy and should be your daily tool. Developed by Fritz Zernike, who won the Nobel Prize for it in 1953, it exploits the fact that light travels at different speeds through different materials, a property called refractive index. When light passes through a cell, it slows down relative to the surrounding media. Different intracellular structures, the nucleus, cytoplasm, mitochondria, lipid droplets, have different refractive indices, so light passing through each slows down by a slightly different amount. Phase contrast microscopy uses a specialized optical ring to convert these invisible phase shifts into visible differences in brightness and contrast. The result: cells appear crisp, bright, and 3-dimensional. The nucleus is clearly visible as a darker or brighter central structure. Cytoplasmic granules, vacuoles, and organelles become distinguishable. Cell boundaries are sharp and clearly defined. No staining, no fixation, no cell death. Living cells in real time. Why it matters: phase contrast is the only widely accessible microscopy technique that lets you observe living, unstained cells with enough detail to assess their health, morphology, and behavior. It is non-invasive, requires no preparation, and takes seconds to set up. There is no excuse not to use it. Setting up phase contrast correctly. 1. Select the correct phase ring for your objective magnification, typically labeled pH1, pH2, pH3, 2. Center the phase ring using the phase telescope or Bertrand lens. 3. Align the phase annulus, in the condenser, with the phase ring, in the objective. 4. Adjust focus until the halo effect around cells is symmetrical and sharp. 5. Fine-tune the condenser height for optimal contrast. Phase contrast is the cell's version of an MRI, except it costs 1/1000th as much and nobody has to lie still for 45 minutes. Fluorescence microscopy, party mode for scientists. Fluorescence microscopy takes things to the next level. Instead of using ambient light, You excite cells with specific wavelengths of light that cause certain molecules, called fluorophores, to absorb energy and then emit it as light of a longer wavelength, lower energy. That emitted light is what you detect. Two sources of fluorescent signal: fluorescent dyes and stains. DAPI labels DNA blue, propydium iodide labels dead cell DNA red, calcine AM labels live cells green, mitotracker labels mitochondria, and hundreds of others. Genetically encoded fluorescent proteins: GFP, green fluorescent protein, M. cherry, M. cerulean, YFP. Cells are engineered to express these proteins fused to proteins of interest, so you can watch the protein in real time in a living cell. What fluorescence allows you to do? Live, dead discrimination. Live cells fluoresce one color, dead cells another. Viability assays. Quantify the fraction of live cells in your population. Protein localization. Is your protein in the nucleus, cytoplasm, membrane, or mitochondria? Protein-protein interaction. FRET, fluorescence resonance energy transfer, measures molecular proximity. Cell cycle analysis. Label DNA and count cells in G1, S, G2, M phase. Tracking cellular dynamics. Watch proteins move in real time in living cells. Key limitation. Fluorescence microscopy requires either staining, which may kill cells or alter behavior, or genetic engineering, which requires cell line modification. It also requires more specialized equipment, a fluorescence lamp or laser source, fluorescence filter sets, and a camera sensitive to low light emission. For routine daily checks, phase contrast is faster and cheaper. Fluorescence microscopy. Because sometimes Brightfield just isn't dramatic enough, and your cells deserve a proper rave with UV lighting. When you sit down at the microscope, you are not just looking, you are systematically assessing 4 critical parameters. Think of yourself as a doctor doing a physical examination. You have a checklist, and each item on it tells you something specific about the state of your culture. Assessment 1. Confluency. Confluency is the percentage of the flask surface that is covered by cells. It is the single most important number you need to know before making any decision about your culture. Get this wrong and everything downstream is compromised. Take a look on this confluency scale. How to estimate confluency accurately. Begin with the lowest magnification objective, 4x or 10x, to get a broad view. Scan systematically. Center of flask, then all 4 quadrants, then edge regions. Estimate each field independently, then average across at least 5 fields. Remember that edge regions near the walls are often less confluent than the center. Record your estimate with a timestamp and, ideally, an image. The 10x thumbnail method. At 10x magnification, cells should fill roughly 70 to 80% of your field of view when you are ready to passage. This is easier to judge than counting individual cells. Train your eye by looking at the same cell line repeatedly at known time points. Within a week, you will be estimating to within 10% accuracy without counting. Checking confluency from one field of view is like checking if a stadium is full by peeking through one door. Technically an observation, objectively misleading. Assessment 2. Cell morphology. Morphology means the shape, size, and structural characteristics of your cells. Different cell types have characteristic morphologies, and deviations from that characteristic shape are usually the first sign that something is wrong, often before cells die, before pH changes, sometimes even before any biochemical marker turns positive. How to assess morphology. Compare to reference images of your specific cell line at similar confluency. Look for consistency. All cells should look roughly similar to each other. Note any cells with obviously different shape than their neighbors. Outliers are informative. Track changes over time. Gradually changing morphology often means slowly deteriorating conditions. Check cell size. Unusually small or large cells can indicate passage number problems or genetic instability. Assessment 3. Signs of cellular stress. Stressed cells are not dead cells, they are cells telling you that something is wrong. This is actually good news, because it gives you a window to intervene before you lose your culture entirely. Recognizing stress early is one of the most valuable skills in cell culture. Common causes of cellular stress to investigate. Media pH. Yellow media means acidic, Too many cells for the volume. Purple means basic. Evaporation or CO2 problem. CO2 percentage. Incorrect bicarbonate buffering rapidly shifts pH. Temperature fluctuation. Incubator door left open or thermal cycling. Serum lot change. New serum lots can have dramatically different growth factor content. Mycoplasma contamination. Often slows growth and changes morphology before obvious signs appear. Excessive passage number. Cells accumulate genetic changes and gradually lose normal behavior. Suboptimal seeding density. Too sparse causes growth arrest, too dense causes contact inhibition. The 24-hour rule. If you see early stress signs, do not immediately panic and passage or discard. First, document what you see with a photo and timestamp. Then investigate one variable at a time. Check pH. Check CO2 readout on the incubator. Verify temperature. Give the cells 24 hours after correcting any identified issue to see if they recover. Stress signs often reverse rapidly once conditions are corrected. Assessment 4. Signs of cell death. Dead and dying cells have a distinctive appearance that becomes easy to recognize with practice. Knowing the difference between a cell that has just died and one that has been dead for hours matters. Fresh death can sometimes be explained by a specific event, brief media deprivation, while widespread old cell death suggests a more serious ongoing problem. What dead and dying cells look like? Shrunken cell body. Cells get smaller as they lose cytoplasmic volume. Very dark, grainy appearance. Increased optical density due to chromatin condensation and organelle breakdown. Loss of attachment. Cells detach from the surface and float in suspension, the floating debris you see in phase contrast. Nuclear fragmentation. In late apoptosis, the nucleus breaks into several smaller, distinct pieces called apoptotic bodies. Complete loss of defined borders. Necrotic cells swell and rupture, losing their membrane integrity. Assessment 5. Identifying contamination. Contamination is the nightmare scenario of cell culture. It can wipe out months of work in 24 to 48 hours, and it is almost always preventable. The microscope is your first and most important contamination detection tool. Types of contamination and their microscopic appearance. The mycoplasma warning. Mycoplasma is the silent killer of cell cultures. At 0.3 to 0.8 micrometers, it is far below the resolution of a light microscope. Your culture can look completely normal while 10 to 90% of the cells are infected. Mycoplasma slows growth, alters gene expression, affects metabolism, and corrupts experimental results, all without showing any visible signs under phase contrast. Test for mycoplasma using PCR or a fluorescence-based assay when you first receive cells and every 3 to 6 months thereafter. These are the errors that make experienced cell biologists quietly facepalm when they see them. Each one is preventable once you know about it, read them, internalize them, and then never make them. Mistake 1. Judging confluency from a single field of view. You peek at the center of the flask. It looks 75% confluent. You declare the cells ready to passage and move on. Meanwhile, the flask edges are at 30% confluency, 2 quadrants are at 50%, and your actual average is somewhere around 55%. You've just passaged cells that needed another day. Why this happens? The center of a flask tends to have higher confluency than the edges, because cells settle toward the center during initial plating. Microscope fields of view at 10x cover only about 1 to 2 square millimeters. A T75 flask has 75 square centimeters of surface area. One field of view samples roughly 0.002% of the total flask area. The fix. Systematically scan at least 5 to 8 fields. Start with the center, then move to each corner quadrant, then check the edge regions. Average your estimates mentally. The whole process takes 60 to 90 seconds and dramatically improves accuracy. Think of yourself as a census taker, not a tourist. Mistake 2. Not documenting microscopy images. 3 months into a project, a collaborator asks you what passage number your cells were when they showed that unusual phenotype, and what they looked like at the time. You flip through your lab notebook. It says, cells looked okay. That is the scientific equivalent of describing a car accident as vehicles were present. What good documentation looks like. Date and time of observation. Cell line name and passage number. Flask ID or experiment code, confluency estimate, percent, morphology notes, normal, abnormal, specific observations, media condition, color, clarity, any unusual appearance, action taken, fed, passaged, discarded, no action, photomicrograph with scale bar and objective magnification noted. The fix most modern inverted microscopes have connected computers or cameras with annotation software. Take a 10x Brightfield and a 10x phase contrast image every time you assess your cells. Save them with a file name that includes the date, cell line, and passage number. This takes 2 minutes. It has saved thousands of experiments when unexpected results needed retrospective investigation. Mistake 3. Passaging at 100% confluency. You had a busy week. Life happened. Your cells hit 100% confluency. You figure you will just passage them now. Surely more cells means a better yield, right? Wrong. Here's what actually happens at 100% confluency. Contact inhibition is triggered when cells touch each other on all sides, a normal biological signal that tells cells to stop dividing. Growth arrest ensues. Cells stop in G0, G1 phase and become quiescent. Gene expression changes. Confluent cells often upregulate stress response genes and down-regulate proliferation markers. Metabolism changes. The glucose consumption and pH buffering requirements of a confluent culture are completely different from an actively growing one. For contact-inhibited cell types, primary cells, some tumor lines, this state may be very difficult to reverse, and cells may enter permanent senescence. The fix: passage at 70 to 80% confluency. This is the sweet spot where cells are actively growing, in log phase, have not yet experienced growth arrest, and will respond well to trypsinization and replating. If your schedule makes this difficult, consider passaging slightly earlier, 60 to 70%, on Fridays rather than risking 100% over the weekend. Some cell lines tolerate confluence better than others. Know your cells. Mistake 4. Confusing cellular debris with bacterial contamination, or vice versa. You look at your cells and see small, dark particles floating around. Your heart rate spikes. Contamination. You throw the flask, disinfect the hood, and file an incident report. Except those were just dead cell fragments, apoptotic bodies shed during normal culture turnover. The reverse error is more dangerous. You see small particles but assume they are debris, when they are actually bacterial contamination in its very early stage. The culture gets returned to the incubator, and 12 hours later, you open the incubator to find every flask in the incubator now turbid and contaminated. The fix: the 24-hour incubation test. If you are genuinely uncertain whether small particles are debris or early contamination, take a 1 milliliter sample of the media and incubate it at 37 C for 24 hours, then examine it at high magnification. If the particle count has multiplied dramatically, it was contamination. If it is similar or lower, it was debris. While waiting, keep the suspect flask in a separate incubator or quarantine location. Where microscopy is going: AI-powered cell analysis. Everything you have learned in this episode, confluency estimation, morphology assessment, contamination detection, health status scoring, is currently being automated using artificial intelligence in pharmaceutical and biotech laboratories around the world. What this means for you as a cell culture scientist. There is a crucial implication here that many students miss. AI systems are only as trustworthy as the person who understands their output. An AI that reports 72% confluency, healthy morphology, is only useful if the scientist receiving that report understands what 72% confluency means, what healthy morphology looks like, and what to do with that information. You need to validate AI outputs against your own microscopy observations, at least until you trust the system for your specific cell line. You need to investigate when the AI disagrees with your eye. Sometimes the AI catches something you missed. Sometimes the AI makes an error in unusual conditions. You need to understand the failure modes. AI confluence algorithms struggle with 3D structures, floating cells, and debris that confounds segmentation. You need to interpret the results in biological context. The AI reports numbers, you make decisions. Current industry applications: high-throughput drug screening. AI scores the viability of cells after drug treatment across thousands of wells per day. Cell therapy manufacturing. Automated QC of CAR T cells and stem cells before patient infusion, too important to rely on intermittent human checks. Bioreactor monitoring. Real-time imaging and AI analysis of suspension cells in bioreactors for biologics production. Colony picking. AI identifies and selects individual colonies with desired morphological characteristics for clonal selection. Contamination detection. Image-based systems trained to flag turbidity and unusual particle patterns before a human operator notices. Summary. What you now know: one-phase contrast is your daily tool. No staining, no fixation, no cell death. Phase contrast microscopy gives you real-time, high-contrast images of living, unstained cells. It reveals the nucleus, cytoplasm, membrane morphology, and intracellular structure in ways that Brightfield simply cannot. Make it non-negotiable. Every flask check should include a phase contrast assessment. 2. Confluency, morphology, and granularity tell the story. Cells communicate their status through appearance. Flat, spread, crisp-bordered cells with visible nuclei are happy cells. Rounded, granular, vacuolated cells are stressed cells. Dark, shrunken, detached cells are dead cells. Tiny, uniform, rapidly multiplying particles are a contamination emergency. Learning this visual language is the single most transferable skill in cell culture. 3. Document every observation. Good science is recorded science. A microscopy check that is not documented is information that does not exist. Photograph your cells, timestamp the images, note the passage number, and write down what you observed and what action you took. 6 months from now, when something unexpected happens in your data, those records will be the difference between understanding what happened and never knowing. If you found this episode useful, don't forget to subscribe our channel. Coming up, Episode 6, Cryopreservation. Your cells cannot stay in culture forever. Eventually, you need to freeze them, creating a living library you can draw from for months or years. Episode 6 covers everything: the biophysics of freezing and thawing, how to prepare cells for cryopreservation, the right cryoprotectant concentrations, controlled rate cooling, liquid nitrogen storage protocols, and the art of successfully resurrecting frozen cells. See you there. Don't forget to like, share, and leave the comment below. We'll see you in the next episode. Thank you for watching.